Hello, everyone. Good day, and thanks a lot for the invitation and the opportunity to speak here. I'm here with my colleague Fabrizio, and we will talk about accelerating drug discovery with, believe it or not, with NIME. And first of all, let's start who we are and who we are in terms of a company. We are a CRO, a contract research organization, and we provide services in drug discovery and drug development for our partners. Our partners are biotech companies, pharmaceutical companies, academic partners. We do chemical synthesis for them, biological assays. We are founded 2007 in Krakow. Krakow is our main hub. We are also in Croatia, in Zagreb, where we have labs. We have offices all around the world. And we have about 900 employees that are working for customers, a lot of scientists, like 40% with a PhD level. And then, who are we? So maybe I start with myself. My name is Jörg Wichert. I'm a physicist by training. I'm working in AI and drug discovery more than 20 years. A good part here in Berlin. I started at Pharma Sharing. It was a charity at FMP Berlin, and last but not least, more than 10 years at Bayer. And 3 years ago, I entered Cellvita in Poland. I'm leading the machine learning and AI team. And with my team are great colleagues like my colleague Fabrizio, who will introduce himself.
Thank you. So my background is in statistics, and I've been working in machine learning and data science for over 7 years now. And I joined Cellvita in the infancy of the AI team 3.5 years ago.
So our talk has two parts. I will start with the first part, that is machine learning model deployment in NIME. And let's start with some common things. So what do you want to do? How can we make our Python-based machine learning models for QSAR and ATME? I will tell you in a second what this is, to our colleagues in the lab and the project teams. And these are chemists, biologists. They don't want to see any kind of a Python prompt or something like this. They want to have something that is intuitive. Well, the solution is easy. Easy in the way that since we have NIME, we use NIME as a NIME server, as a front end, providing an API for our internal customers that are our colleagues, so that they can use our models, put their structures there, and get the predictions, and that's it. So, let's say the value of this approach is that we have a unique platform for making our models available to our colleagues, and we have, let's say, an easy, maintainable platform for us. So let me start with the setting. So QSAR, what is QSAR? QSAR is the abbreviation for quantitative structure activity relation, and it is the art to predict the outcome of an experiment having only the structure of the compound. Why only the structure? You can say you can do the experiment. Yes, but doing the experiment means you have to synthesize the compound, right? And we don't want to synthesize the compound to make the experiment. We want to have a prediction that predicts the outcome of an experiment before we synthesize it. So we can sort out the good guys and synthesize them, and sort out the bad guys and forget about them. So maybe you have seen this molecule before. If you don't recognize it, stay tuned. I will uncover it soon. And we can ask, what is the solubility in water of this compound? And the answer is, well, it's about 3 grams per liter. That's not a lot, but it's enough at room temperature. And this is a classical question of QSAR, right? You provide the structure, you ask a question, and you get the answer, and this is called quantitative structure-activity relation. So why are we doing that? Because we want to predict what is called ATME, that is the fate of a compound in the human body. So that is about absorption, distribution, metabolism, excretion, and toxicity. And it tells us how the drug is doing the journey through the body, and we want to know this. And the more we can predict, the less experiments we have to do, the faster we are, the more effective we are, and the more money we can save by predicting things that we can sort out. For doing QSAR, we need the holy trinity. That is, we need data, we need descriptors, and we need algorithms. And this is where the machine learning comes in, right? The data, that is something that comes from public domain sources, from in-house sources, from our clients, and we can do a lot of data processing in KNIME. I will show you a pipeline soon. The model development, is done internally in Python, because Python is the lingua franca of machine learning right now. So everything is built in Python. It runs in Docker containers. We use scikit-learn, RDKit, and we do the whole versioning in MLflow. So this is happening behind the curtains, and we use KNIME to be the stage of this, and we do the whole model deployment on the KNIME server, and we have the KNIME server as an API for our colleagues. And here's the typical QSAR workflow for data processing for the solubility data. So you can see in the items are a little bit outdated because we are still on KNIME server, and that uses KNIME 4.2 or something, but we are moving to the high KNIME hub soon. So we take the data from the Aquasol database, we do some some preparation of the compounds, we sort out some stuff, we normalize the structures, we standardize the structures, we can use all the RDKit canonical SMILES nodes that we have here. There are other nodes from LADA, from Genitech, from ZDK, so commercial nodes from ChemAxon. This is all there, and more, let's say, chem informatics companies are providing the structures, right? And their nodes to run them in KNIME. That's great for us. I mentioned it. In MLflow, we are doing the versioning of our models. So here is the solubility model I was talking about. This is all in MLflow, and in MLflow we are storing our models so that we can access them in KNIME. And here is a little bit the heart of the story. Right? This is the workflow for the model deployment. It's the model template. We write the model description here. We do some processing here, and this was done by my colleague, and he says, okay, dear Jörg, if you want to have a new model, open this, and this is where the magic happens. Here I connect with a call. This is a bit of Python magic, right? I take the model from the server, and then the whole interaction works on KNIME. And this is what our colleagues will see at the end. This is the API from the KNIME server about aqua solubility. If we push the button, run, we first get a model description. So this is something that we have for our internal colleagues. It tells us, okay, general information, water solubility is very, very important. This model is built on support vector machines and some machine learning mambo-wambo. We use this kind of descriptors. This is the data set. This is the performance you can expect. If you want to know more, read it. Most of the people don't read it, so the button to go is there. Then they go. Later on, they ask me, oh, what is the model doing? Well, you have seen it. Well, the model description was there. No. They want to have their result, so they put the results, comes on the next page. Here goes, enter your data, input SMILES. SMILES is a format for storing molecules. And here comes the output. That is the solubility of the molecule you have seen before. It's ASS. That stands for acetylsalicylsäure. And we know it as aspirin, right? So this is the water solubility of aspirin in log S units. It tells us also, oh, there is a similar molecule in our database. So the model has seen this already. It tells the similarity is 1. So we know about the solubility, so the model does not predict it. It takes it from the database that you can see there. And then the user can download the data, the result, and that's it. And if he has any questions, of course, there is somewhere a button, please ask one of my team members. Usually now I have to do it, right? And then that's it. And this is how we deploy our machine learning models in KNIME. Very nice platform. And as I said, this is the server. The hub is coming soon. Right. And now comes the second part of the story, and I hand it over to Fabrizio. Thanks.
Thank you. For my side, I will instead talk about a different experience. We tried to improve some processes that were happening with older software inside Cellvita, and we wanted to add some automation and some more custom features to the processes that were being performed. And we tried to do this with KNIME. Specifically, what we did was LIMS, which is a laboratory information management system. First of all, let's talk about what the problem is and what we are trying to improve. We have an analytical pipeline in Cellvita. Our chemists produce several reactions every day, and they have to check if the desired product is actually present inside the final sample. They can do some preliminary tests on their own, but if they want to do some advanced analysis, they need to talk to specialists. And for this, they have to go to our analytical team. Considering the size of Cellvita with hundreds of chemists and double at least the amount of reactions every day, this means that there is a significant overhead in the communication between the two teams. Specifically, when a sample is sent to the analytical team, they have to record somewhere where it comes from, what is the priority, what is the type of analysis that is to be performed. And these are the kind of operations that are done through LIMS. So let's talk about what are the functions of this software. The first one is sample registration. Sample registration includes, of course, just writing down the name of the sample, the name of the scientist that require the analysis, the type of analysis, and the structure that should be extracted. In our case, we also include some preliminary analysis results that are performed by the chemist. So there are some PDF files that are uploaded. These are liquid chromatographies. And we include inside our KNIME workflow also some pre-processing of the structures that extract some, let's say, key features, like the solubility that Jörg was talking about earlier, which is very useful for our purification analysts to know about. In second order, we need to follow the project. So there has to be some sort of assignment of the sample to a chemist, to a specialist. There has to be a way of communicating what is going on. If the status changes or if there are some problems, there needs to be a direct connection between the chemist and the specialist. We need to log the operations, so what is happening right now, what has been already done, and what is the next step. We need to produce a report, which is when once, of course, the analysis is finished, what is the outcome, how it's going to be communicated, and finally, we have to actually send this report and communicate the end of the analysis. So, before thinking about doing it in KNIME, and in general, I would suggest everyone to think about all these questions. When you develop a software, you first have to ask yourself how it should be deployed. How many users do you have? How many concurrent users? How many requests do you expect to receive? How many times the communication has to go back and forth, and which kind of files are exchanged? Maybe some are too heavy to be sent in a normal way, or they should be progressively purged because they're going to fill up your data storage. Is there sensitive information? This is a very important one, of course. Every time you send information back and forth, can I afford to have it on the cloud? Should it be always local? Should it be anonymized? Which outcomes can, and this is very specific for us, of course, which outcomes can it have? How many solutions should there be? Is it only positive and negative? Could there be also extra questions? Could it be, I need more sample? Many things. And finally, something a bit more hardware related. How are the final users going to access your software? Are they all on the same system? Are some of them using command line? Some of them like to use web browsers. Some of them the computer is, something esoteric. I want to just move the mouse, and that's the maximum that I want to do. And since the last thing is quite true in our company, KNIME is a great option. So as you can see here, we used mainly two softwares, the omnipresent and honestly very good Microsoft Excel for report generation and KNIME for the other 4 options. This is because we didn't want to change what was already working and what would have made the protocols maybe a bit more difficult to, let's say, update for our teams, right? Sometimes they are happy with what is working, and you don't have to change it. You change around it. Let's talk then about how KNIME helped us solve some problems that were tied to this definition of the software requirements. I will go relatively quickly over these ones because there's a lot, but of course we have the question and answer if you want to know more specifics about them. Okay. So the first one is, in the input phase, we want to have a dynamical user interface. Different analysis, different samples require different specifications about them. In our case, we are talking about chemical procedures, so maybe they require different treatments for heat, or we don't care about it because it's always performed at room temperature, or maybe we need to know more about the sample that you're sending because it's advanced analysis and it requires more files, like you can see here. When we change from LCMS to an SFC, more files are required because it's a more complex analysis that needs more information. And KNIME is very good about this because we have components, we have our dynamical re-execution every time that a single option or a Boolean is selected. And what we could do was also rerun some Q calculations. So every time they change the site or the analysis type, it would immediately go back and recalculate how much time is expected to pass before they can get an answer. You can see here a very wiggly representation of how it is under, but you can see all the case and switches and all the re-execution nodes. We are also interested, of course, in reducing, and this is in general in computing all the time, reducing redundancy. Every time you write code that is using a function that you already have defined, you should not rewrite the function. You should try to keep a library of them. And we have seen also this in the workshop, the L3. And what we do here, we do it with components and the nested components. So we know that some analyses have shared information that has to be passed and share operations that are done. And so we keep them together. But these are small parts that are then joined together into components. Since, of course, we are the AI team, we also want to try and give some predictions, not just automate the service, but also help them to become more efficient. And we try to do this by taking all the past informations that we start gathering by creating these limbs. We save all the past operations into an ELN, which is an electronic lab notebook. And we take the success and failures, which luckily are actually quite rare, and we create a new predictive model that is capable of suggesting new ways of performing or old ways of performing the analysis that are more likely to be successful. Sometimes these are just based on rules, which is totally fine, and other times we try to do some machine learning with good results. We want to show a queue, of course. As you've seen earlier in the project management, when I was talking about it, we want the person that asked for a request to know how much time is going to pass before they have the answer. And KNIME is very good about this because it has dynamical tables. We can download from SQL. We created our own data storage in a safe disk, which is separate from where normally people can access. Only our team and KNIME server itself can access that space with the information that has been anonymized already. as you can see. And it's then passed, ordered, and can be visualized. It works. For communication, we decided to use emails. This is due to the fact that it's already an ubiquitous way of communicating information in our company, and I guess in every company. But in our case, the protocol for most communication between chemists was already emails, so they are ready to check them quite often. For example, if someone is using some dangerous reactants, they should tell the whole floor that there is a risk, possible risk, so everyone is constantly checking for these things, and now they also check what is the status, if there's a change in the queue, or if something is finished, and they can go and take the results. This works also for priority, which has to be authorized by a manager, and it's everything handled automatically by KNIME. They can ask for priority, and boom, it will find who their manager is, send the email, and they will have to approve it before it is out. We talked about what are the possible outputs, what are the possible conclusion states. In our case, we have only 3, which is success, failure, or on hold. I need more information, or I need to talk about what is happening. And we do everything through KNIME once again, with uploading of reports. We can also extract more information from the queue. This is for the analyst if they need to remember something. You can see here. This is a cute trick that actually I think maybe not everyone knows, but it was talked about in the L3 workshop again. If one puts an infinite loop at the beginning and at the end of a workflow, you will have the operation continuously go forward, and it doesn't require the person to open a second workflow to do the same operation. Quite cute. Finally, what happens all the time? There's going to be errors. This is not about workflow errors. This is about human errors, which are unfortunately inevitable. So what we want to do is create a way to not have the workflow crash or the analysis stopped, but instead to stop the request or maybe just warn the person that is asking. And what we do is we use a Python script that selects all the possible informations that have been given wrongly. And after that, they can be categorized in 2 ways. So what you said, what you gave us was an acceptable input, and in that case, we have a blocking issue, as you see there. Or what you said is just something that requires a warning. And it will not block you, it will just make something extra appear, and it will say like, okay, hey, be careful, something happened here. Are you sure that you want to go forward? We will have to set something as default. Otherwise, there will be an error, and they can simply go back and change and specify, and you can also write several messages, one after the other. So, in summary, I have to say that the implementation of our machine learning models in KNIME has been quite straightforward. We have an integration with APIs thanks to the nodes, and we are able to deploy them and have our chemists and medchems and biologists use them. And in the sense of optimizing operations and processes through KNIME, I would say that this was almost revolutionary. My background is in statistics with a little bit of programming that then I honed over time, of course, as I worked, but I am not a front-end engineer, and it would have taken me months to develop something like what KNIME made me do in 2 weeks as a prototype and 2 months as a finished product, with a UI that people like, with warnings, with looping messages and everything. The interface with emails as well was a great tool. So we also validate users. We keep everything in-house because KNIME Server, as you know, of course, can be put in your own company. And in the end, I would say that this is a great success story that we managed to obtain thanks to KNIME itself. With that said, thank you for your attention.
Thank you very much, Fabricio and Jörg. I want to push you a little bit to the center of the stage as we have a couple of minutes still for some questions. So I want to open it up to the floor to see if there's... I see you there at the back is already a hand raised. Just want to scan as well as the mic goes there. Please go for it.
No, it's not working though.
All right, let's go to another question taker as we sort out the mic there. I think we have another question on this side of the room. Please go ahead.
That one is not working either.
Now you can hear me?
Yes.
Okay.
All right.
Go ahead.
Let's go this side. Yeah.
A question that came up online, which is probably like, what data processing are you doing with KNIME when you're getting the molecules in? What's that exactly? Can you maybe give a few words there?
What we do is data cleaning. We remove molecules that contain things that are not, let's say, interpretable. Sometimes you have these R groups, and you cannot calculate anything, so they are sorted out. Then anything that is not an organic molecule can be filtered out. Then we inspect it, because some things are non-organic, but still in the pharmaceutical industry used. So these kind of things. And we have seen these two nodes that are Cellvita nodes, and below that is a kind of Chemaxon script running some checkups. Are the valences correct? Are the, let's say, is it shown in the right way? So because sometimes people are producing malicious structures, I would say. So and this is all sorted out so that the model can interpret it.
Thank you very much. Let's go to the second question we had.
Yeah, thank you so much. It's a very impressive effort. Can you build on this generative AI to predict a new molecule using the already available molecules?
Yes, yes. We are trying now to push this forward. There's a lot of generative AI for chemistry out. You can access it and see the GitHub. Some is good, some is bad. Unfortunately, it does not work as nice as an LLM, like please design me the next kinase inhibitor that is super selective. That does not work. But
Yet.
Not yet. There are some, let's say, good approaches, and we are experimenting with this, but we have not shown it now.
Yeah, we actually have already one that is dedicated to part of the molecule, not generation of the whole molecule, but part of it, and that one is already working. But generation of the whole molecule is a bit more complex.
Okay. Are there any reasons behind why it is not yet giving the best results, like LLM?
Because it actually doesn't work for other things either. It's just that humans see script and they think that it sounds human.
Okay, thank you.
Thank you. I think we had one more question on this side we still have time for.
Yes, that's another question that came in online, which is, in their experience, it's often very difficult to get customers comfortable with training machine learning models on their proprietary data. How do you approach this challenge with your customers? Could you maybe comment on that?
Oh, that's a very good question. Though usually we are, let's say, completely focused on our customers' needs. So when they say they want us to use their data exclusively, then we do that. We can use then data from the public domain. We cannot, since the data belongs to our customers, we cannot mix it up with other customers' data. So we have a kind of silo for the data. But as long as we don't distribute, let's say, the information or do any kind of, what's it called, contamination, right? There is a way, let's say, to learn the features of the molecule without sharing the IP-protected part of the structure. So there are there's a federated learning paper out recently that we did with the colleagues from LASA, who are also KNIME partners. They have LASA nodes here. And there's a lot of things done, because more and more people want to build really cool models, but they don't want to share their IP. And there are now ways and initiatives to share the knowledge without sharing the protected part of it. Sounds a little bit weird, but it works.